Grande do Sul, Porto Alegre, Brazil. She also has a master's 2011 and a PhD 2015 in geosciences from that university. During that time down there, while she was studying and working on degrees, she was assistant at the Paleontological Museum, involved with management of their paleobotany collection. Her major research interests, which will not be a surprise given the topic tonight, involve paleobiology, paleobotany, paleoecology, and the fossil record of plant-insect-herbivore interactions, which she will be talking about here this evening. Multiple various awards and research grants, and an impressive list of publications and abstracts, and not surprisingly, she's a member of multiple professional membership organizations, GSA, Paleontological Society, Brazilian Society of Paleontology, Paleobotany and Palynology, Latin America Association. So without further ado, please join me in welcoming Esther Pinheiro.
Thank you, John. Let's see how I do with the microphone. So yes, I'm Esther. I'm from Brazil. You can see by my accent. I'm a paleobotanist, and I work more specifically with plant-insect interactions. How insects used to eat plants in the geological time. My PhD thesis was about Permian age and Gondwana, how the herbivory was distributed through Permian and along the all Gondwana. And then this came on me like lots of questions, like these patterns that I'm seeing so old, so long time ago, we still can see it, how this fits in a major picture of geological time. So I contacted Ellen Curano and asked if I could work with her and learn more about angiosperms and more modern, I can speak, fossils and plant fossils. And this is what I can do here in Wyoming, work with paleoreconstruction, food webs, herbivory in Paleogene here from Wyoming, because you live in a spectacular place to work with Paleogene. You have the best fossils from PTM, as we will see further. And so, yeah, today I will speak about how we reconstruct forests, food webs, and climate using plants from Wyoming. Okay? Moving on. So why Paleogene? Why Paleogene is so important? Because studies shows like that during Paleogene we have two really important events: the PETM, the Paleocene thermal maximum, and ECO, the Eocene climate optimum, that were really, really hot periods in time. And we know that during this period we have no ice in the poles and really high temperatures. And what this means that probably we didn't have a gradient of temperature as we have today. So it was more homogeneous distribution of temperature, and probably we would assume that more homogeneous distribution of life. So this is why it's important to study Paleogene, because with Paleogene, you can use this data from Paleogene to predict the future global warming that you see. I saw in a couple weeks, next weeks, how global warming is changing our ecosystems. So with this data from Paleogene, we can modeling and see how food webs, how plant structure, and how the climate will change in the next future. When I talk about Paleogene, I'm talking about Paleocene and Early Eocene. I'm sorry. And so from 66 to 48 million years ago. In this talk today, I'll be more specifically, I'll talk just about Early Eocene, that our most update data. Okay? So, And why interactions? Why is it important to study food webs, not just paleoclimate or not just the forest? Because we know that biodiversity is really, really close to the richness of interactions that we have between species. So with as much species interacting and like eating each other or use eggs like a bird pollinizing a plant, or even like a fungal, all this kind of interactions increase biodiversity and open new resource and new niches to new species to exist. So it's really important to maintain biodiversity. To maintain biodiversity, we should maintain interactions, all the food webs that we have. And when we talk about biodiversity on Earth, we are primarily talking about terrestrial plants and arthropods. They are the mega diversity groups that we have on land now. So when we study the interactions between these two groups, we are studying like more than 50% of biodiversity on Earth. So they are really, really important. So plants and animals, more specifically arthropods, can interact in uncountable ways, like as a shelter, like insects can use a plant as a shelter, as a reproduction. So insects can put eggs in the plants, in the leaves, for example. Oh, and also the plant, as I told you, use insects to reproduce. When a bee goes from a flower to other flowers, helping plants in the reproduction. And as food, insects, of course, can eat plants. And for us in paleontology, the most common and the most important usually is the record of how insects eat the plants. Because when the, sorry. I always have to do this, doesn't matter. Okay, so, sorry. As you can see here, this is an example of a modern mine. Mine is when an insect put the egg inside the leaf, and the arthropod develop all inside the leaf. So when the animal starts to walk and eat, it does this tunnel, and the plant reacts. It has a physiological response and does this different coloration, the different tissue. And this different coloration, it stays in the fossil record. So as you can see here, this is a Glossopteris from Permian, and you can see this reaction tissue, that more reddish. That's the same idea that you see in modern plants. This is a hole in a modern plant, and this is a margin feeding. This picture, for example, I took last week in the Smithsonian Botanical Gardens. So, as you can see, it doesn't matter what age you are working, you can see the tissue in the plant. So, since Permian and modern is the same kind of physiological response. So, we can follow this through time. So, in Wyoming, as I told you, Wyoming is amazing geologically speaking, other aspects too. But when we talk about Paleogene, we have several basins, not just across Wyoming, but just all across the Rock Mountains. And this presentation that I'm showing you today, it's part of a bigger project of Dr. Purana, that she wants to see all the geographical distribution of plants during Paleogene, and since Paleocene until the end of Eocene. So we have since one basin that is really new data that we have, and now I will show you today about Wind River Basin, Bighorn Basin, and we will also have people working in Hanna Basin. And I think in a close future we'll have a better view of what is happening during Paleocene with the flora here in the Rockies. So our goals today is to reconstruct early Eocene flora, to reconstruct early Eocene climate, and see how the difference in the plant community are influencing herbivory during early Eocene here in Wyoming, And when I'm talking Wyoming today, I'm talking more specifically about Pinheiro Basin and Bighorn Basin. So, what we know? Pinheiro Basin, as you could see, it's really close to here actually. It's in central Wyoming. About the flora from Eocene, we have just one publication from 74 from Dr. McGinnity, and it's a beautiful work. He described all the morphotype of plants and all the pollen of plants, and oops, again, oh my God, seriously. Yeah. I did block this, but this did not push. He described 36 species of dicots, that is our goal today, and he concluded that we have like a tropical kind of flora here in Wyoming during this time. The other basin, as I told you, is Bighorn Basin, and we have several papers about the Bighorn Basin flora, because there we have the record of BETM, was what the first place that we have like terrestrial record of BETM. But today, more specifically, I will work with this paper from Dr. Corano, that she's comparing herbivory in Bighorn Basin, different sites. And I work more specifically with this site that is also early Eocene and matches really well with the Pinheiro Basin, and can do really good comparisons and see what's going on as a whole. So I not give you much details now. So again, so we are working these two basins, Bighorn and Pinheiro. This is a color code that I use all around my presentation. So red is always be the margin sites of Pinheiro Basin. Black is always be the central, the interior sites of Pinheiro Basin. And blue are the sites in the interior of Bighorn Basin. So you have here I want to show you like a perspective where it's in the basin, like Bighorn interior and Pinheiro interior and Bighorn, they are more inside the basin, and maybe because of that we are expecting that we have more similar results. We will see if we have or not. And the Pinheiro edge. Okay, so let's talk about geology of these places. So Pinheiro, they have a really distinct kind of physiognomy now. Like this is in Shoshone Forest, close to Du Bois. You can see that is a forest, and we have these layers of like really a thin kind of sediments made by volcano. So you also can see ashes. They belong to the A-cross formation. They have a age from 48 to 50 million years ago. And we're working for sublocalities along the same layer. In the interior of the same basin, we were in a different formation. We were in the Pinheiro formation, that more like a floodplain kind of environment, and a little bit older age, and we have like 8 localities. And you can see that today is really different kind of landscapes, much more dry, and this is it's your forest as your yard, practically. And the Bighorn Basin, this wood formation, is also a floodplain, but for more overbank kind, from meandering kind of rivers, also 52 million years old, and we work in 7 localities. So, just an overview. We have 3 different areas, that Pinheiro edge, Pinheiro interior, and Bighorn. Bighorn is interior too. They have more or less similar age, but the interior and Bighorn, they are 52 million years old, and the edge is a little bit younger, it's 49. So maybe this 2 or 3 million years of difference it's important, or it's not, I will show you today, but when we start to work, this was a question, this age makes different or not? We can all compare, we can all put together. Because when I was working Permian, 3 million years, it's absolutely nothing. We don't have this kind of resolution. So for me, it was like, seriously, 3 million years? And she was like, yes, Esther, pay attention. So let's see. Okay, this is just to show you how we work. This is actually me in the Shoshone forest. I brought today some hand samples, and this is more or less how we do it. We get big blocks, we try to split, because this is really far from the car, and we have to carry all the rocks. So yeah, and I'm short. So we count when we are in the field what I'm able to identify there, and what I'm not, what has like traces of herbivory that I think is important, that I cannot identify actually in the field. We split in smaller pieces, and we wrap in toilet paper, yes, toilet paper, and we bring to the lab. We call these fossils wrapped in toilet paper mummies, because like, yeah. We try to have fun, you know. So when we get to the lab, I have all these rocks, and I know they're plants, but they need to have a name. So by modern botanical rules, nomenclature rules, we cannot give a plant name if you don't have the flower and the fruit. And usually we don't. We don't have flowers connected to leaves or connected to trunks. So we use morphotypes. We call morphotypes. You use this guy more specifically, the Manual of Leaf Architecture. That is really, really simple. We get impression of a plant. I know it's dark, you cannot see. But first question is like, has a teeth or does not have teeth? How is the primary venation? Is pinnate or palmate? How is the secondary venation? How is the tertiary venation? So we can separate different morphotypes. Sometimes we are able to give these morphotypes names, as you see later. Here we have a type of populus that we have until now. That is the popular aspen. But sometimes you can't. So here I have one leaf that I gave name tooth in the apse. This is the name of my morphotype. So it's not always that it's possible to give her actual name. Actually, this is a project that we are about to start, that is get all the publications from these age and try to put everything under the same names. So it will help us to do better comparison about sites. Because now, here in Wyoming, it's been relatively easy because Dr. Curano already working almost all basins. So in her lab, we try to give everything the same name. But when you go to other university with other researchers, not necessarily they use the same nomenclature. So this also can be a problem. Not a problem, but we are always aware that sometimes one tree, like for example sassafras, has in the same tree more than one morphology of leaf. So if you have this in the fossil record, probably you have like 3 morphotypes here. So we know we have this problem in our data, but we don't have how to resolve this for now. So usually morphology, just the morphology, is not the most important character. This is why usually we try to look for the tertiary variant, because the tertiaries are more conservative in species, and genetically they are more influenced by genetics and not by the environment as morphology. Morphology has a huge influence by if it's wet or dry and the temperature. So yeah, we always have this problem in our mind. Okay, so now I have my plants, I gave a name, I count, and I want to know how these plants were interacting with the other organisms, for example, arthropods. So we have a guide. We call the, here, let me see. Guide of insects and other insects and other damage on compressed plants. And this guide was like a revolution in our field, because until this guide, we didn't have like uniform kind of description of this damage. So it was really, really hard to compare damage through time and through floras. And then Dr. Labandeira, with collaborators, did this. It's a visual guide. It's like a field guide. You have a picture, a brief description, and you can use, doesn't matter the time, you can use since like Devonian, we have a record of plant-insect interaction, even to modern ecosystem. I can go today to the forest and try to look at from this kind of damage. Sometimes I use like interchangeable damage types, that this is specifically, like the morphology. Sometimes I can call traces of herbivory, or or sometimes I call just DTs. That is the abbreviation that we use. Okay, so just one more example, so you have an idea how we do that. We get a leaf and we compare. So here, can you see we have like the reaction tissue that I told you? This is a Duntamopteris from Peruvian, and this is how it's in the guide. So the description is about the arc with less than 180 degrees in the margin. So it matches. This is a plant from Carboniferous actually, but you can use, really doesn't matter what age, doesn't matter who is the host. So it's really, really helpful. Okay. These damage, they are grouped, what we call functional feeding groups. They are like guilds of what these animals eat. So we have like whole feeding, margin feeding, skeletonization, surface feeding, piercing suck, that when the mouth part is like an escalate, is like, have just a little hole, is really neat. Oviposition, that is actually not a type of herbivory, but because leave a damage, leave a mark in the leaf, we study altogether, but oviposition is when insect leaves an egg in the leaf, but does not damage the leaf, by definition. Mine, a gall, is when an insect put the egg inside the leaf, and the larva develop inside the leaf, but when the insect is ready, opens a hole and leaves. That does not stay inside the leaf. Different from the mine. Mine, they stay inside the leaf all the life. Seed predation, pathogen is like fungal, for example, and with boring. This is important because give us idea, a more bigger picture what is going on about food webs. It's like you can differentiate people that is vegetarian from vegan, and even inside vegan you have like different types. You know, everybody eats plants. Yes, everybody eats plants, but you have different ways to eat these plants. So this gives us like also a type of specialization, like a mine and a gall is a really more close type of relationship that have to have like co-evolution between the plant and the arthropod about chemicals and mouth parts than just to do a hole in a leaf. A hole is much easier. Go there, do a hole. This is why we have holes since Devonian. Okay. So, let's start to talk about our results, what we found about the climate. For temperature, we use the margin of the leaves. So we see the proportion in every side, toothed and untoothed leaves, and we put in a regression and we have numbers. So this is why do the morphotyping and the taxonomy really well done, it's important. So we have that all the three areas that we are working, they have more or less similar temperature, around 20 Celsius. And this is really, really warm, even more if you compare with minimum temperature today in Wyoming, it's 8 Celsius. And in early Eocene was 20, so it's a huge difference. It was really, really warm. Same with precipitation. So the precipitation we do comparing the proportion of different size of leaves, the small leaves, medium, and big leaves. And we see also a high precipitation. We have more variety here, but it's still much higher than today. In Eocene, they are all around 100 centimeters, and today is 39 centimeters, the mean annual temperature. So Wyoming, during early Eocene, we know for sure was warmer and hot than today. This, in the museum, I give tours for the kids. Every time that I say that, they were like, seriously? Like, warm? I'm like, yes, really, really warm. It's hard to believe sometimes. About the flora. So, because you know that we cannot collect the same number of samples in every layer that we go. So we have differences here. So in the Pinheiro edge, we collect just around 900 fossils that we could identify as dicots. Now we're talking just about dicots, not conifers, not fir, even not monocots, just dicots in this result. So we have 900 in the Pinheiro edge, 1,700 something in the interior, and 1,801 in Bighorn. And different numbers of morphotypes that were identifying each area. So the edge has 29 morphotypes of dicots, the interior has 38 morphotypes, and the Bighorn has 24 morphotypes. And so I want to show you what I'm talking about. What are these morphotypes? That is the beautiful part of my work. So this is the McGinnitea. It's a genus because of Dr. McGinnitea, as I told you, was the first person that worked in this area. It's a beautiful, huge plant. I have some samples here that you see. They look a little bit different from this one. They have teeth and all these lobes. It's a beautiful and easy to identify kind of morphotype. Dicot tree, as I told you, it's not always we can give a name, so this is like dicot tree. It's an entire margin kind of leaf. It's really, really common in the three areas that we'll see. This is Canavalia. Canavalia is really, really also easy to identify because of this base that is more flat. And the tertiary, they ramify, and it's really easy to follow in the fossil. So also really common to this age in this area. This is one exception that is not a dicot. It's a fern. It's a Lygodium. Really, really, really common. It's beautiful. I count just in Pinheiro edge more than 1,000 leaves of this guy. So yeah, this is why we don't put in the comparison. It's really biased. This is a populus, as I told you, a type of aspen, since he used early Eocene. You can see the teeth. That is really conspicuous for the species, and the pair of veins. As you can see here, these are primary veins too. It's really easy to identify this. And a Symploculus, also really common. In this picture, I don't know if you can see, but the veins do a loop close to the margin. It's really neat and also really, really conspicuous. And these are damages, just to show you again, these are windows, the DT21. Okay, more morphotypes. Platycaria, also really common. They have these teeth, and the vein goes directly to the teeth. It does like a triangle with the teeth. It's also really easy to identify. I have the morphology of the secondaries. Dombert, the teeth. It's really much easier to identify and put morphotype lives with teeth than lives with entire margin. And because the teeth have a really range of morphologies, so helps a lot to put in different categories. And this is almost also with teeth, also very, very common. Okay, so when we compare the composition, the floristic composition among the areas, we can see they are totally different. So the proportions are different, and as I told you, the composition. So here I put Lygodium just to show you how important it was for the Pinheiro Basin. And actually, we barely saw Lygodium and the Bighorn Basin. This means something because Lygodium, even now, is closely related with really wet environments, and not wet about precipitation, but with the availability of water in the soil. So this is telling us that we have lots of water available in this area, in this basin, available for the plants. So to give names and put the perspective. So this is what Lygodium, as I told you. So in the edge, the second most common is dicot tree, and dicot tree is also important in the interior of the Pinheiro Basin, and and in the Bighorn. So just dicot tree and populus are in the top 6 of the 3 areas. So it means that if you go, let's pretend I'm walking the Pinheiro in the Du Bois area in Eocene, I can see one kind of physiognomy, and if I walk 100 kilometers more and I go to the interior of the Pinheiro Basin, and I will see a totally different kind of forest there. This is the main conclusion here. So, okay, I was making that is really important in the edges, not in the others. This is Canavalia, also important just in the edge. Populus, as I told you, it's common in the 3 areas. Platycaria is really, really important in the interior of Pinheiro Basin, and important also in Bighorn. Dombert, and almost, yeah, I think that's it. Simplocus, Dombert, almost. And, as I told you, in the total of the morphotypes that we have, just 10% are shared. Just 10% of all the morphotypes we can see in the interior, in the margin, and in the Bighorn Basin. It's not much. It's really not what we are expecting, since we have all this really similar temperature and similar precipitation between the areas. So these were really, really unexpected. Okay, so this is our donation, it's a PCOA that decides, as I told you, we have this color coding, so Pinheiro edge, Pinheiro interior, and Bighorn. And this is how the sites are similar or different in relation to the floristic composition. It's more or less a more statistical way to show this table. Is to see if this table is statistically significant, different. And it is. So yes, the flora in the Pinheiro edge is different from the interior and from Bighorn, but Bighorn and the interior of Pinheiro has some similarities. And as I told you, these similarities could be because both are interior of the basins, or they have more similar apes. We are still looking for reasons here. Okay? About diversity metrics, so we can see that the most diverse flora is the Pinheiro edge, followed by the Pinheiro interior, and the less diverse flora is the Bighorn. This was really, really unexpected too. And but it's interesting that when you work among sites see that the numbers are similar. And when we go to the side by side in the interior of Pinheiro, we have a more variety of numbers. The diversity changes a lot in the same area, depends where are you digging. So this maybe is related at why it's more similar with Bighorn and not with the data from the same basin. Okay, so what the general view about the flora? So, Pinheiro edge is more diverse and for sure has more surface water available for these plants. Bighorn is less diverse, but more similar with Pinheiro interior. They have more similarities. And maybe, not maybe, yeah, probably, what we are seeing here, it's a kind of endemism. We have like endemic species, endemic distribution of species here in Wyoming during early Eocene. Okay, so let's move on about the plant-insect interactions. So we have different frequencies of herbivory acting here. The Pinheiro edge, just 10% of the plants that we identified had traces of herbivory. So just 10% of the flora was eaten by insects. This is a normal number when you go to Paleocene or to Paleozoic or Mesozoic. And for PETM and ECO, that is more related what we were working, these are more normal kind of rates, like 45% of the flora was eaten in the Pinheiro interior and 55% in Bighorn. This is really high, but it's more, usually is what we find in these really hot kind of ages. Nowadays, depends of a flora. A tropical flora has like 30 or 40% of the flora being eaten by insects. If a more cold kind of forest, like conifer no, but yeah, more alpine kind, you have a rate more close to 10%. And they are eating. They have different rates of herbivory, and they are also eaten differently. So, in the Pinheiro edge, the most frequent kind of damage is the fungal. This is really, really unusual. Usually, as I told you, the most common is holes and margin because it's easier for insects to do it. Fungal, it's not easy to find with the fossil record, and also it's saying to us that this flora, the Pinheiro edge, is really wet. Because to have fungals, even we know that in Wyoming we don't have fungals in our walls because it's dry. So, but if you go to Brazil, oh my God, you have to take care of your house, or the fungals will take care for you. So this is saying that Pinheiro edge was a really wet environment. The other damage are, as I told you, DC2 is a hole feeding. This is what's totally expected. Skeletonization be the most common in the interior. It's not very common, but it also we can work with that. And margin feeding. So this is more or less the distribution of damage in dicots. So, example of damage. This is a dicot tree. Can you see the little circles? Yeah, little circles. All the surface, there are galls. This poor plant was all covered with galls. Probably die really, really soon. Not common. This is a new damage for this area. We don't have a record of this before. This is the fungals that I was talking about. This black blotch that you can see. Super, super common and super, super common. This morphotype in specifically dicot tree. This is a type of hole that is more a window shape, not that circle. Also really common. Here we have a beautiful mine. This is from, I can be wrong, but I think it's from Bighorn Basin. You see the mine. Can you see the path? with the reaction tissue. So, and you can see by the size, the way that the animal, the arthropod was walking, usually it starts more narrow and gets more large. So probably it was walking this thing. And here you can see the edge of this leaf, the galls, these circles. There are galls all over the edge. I can show you here. Okay. Really, really neat. Okay. So we have this more than 30 morphotypes. We have 10% of morphotypes shared between these areas. These same morphotypes in different areas, they are eating the same way. They have the same kind of hostess, the same kind of herbivores are eating these morphotypes. So we did this ordination to try to answer this question. And we see that actually we don't have many difference. We have all the morphotypes are more or less together in relation to damage. They don't differ very much, with 3 exceptions. And this is really, really interesting because when we're like, why this tree specifically, we try to look for answers. We see that these 3 morphotypes are hostess of the most exclusive kind of DTs. Exclusive kind of DTs is a type of DTs that you find in one area and you don't find in the rest of one. So, for example, the dicot tree has 9 of these 10 exclusive morphotypes from Pinheiro edge. So the same happens with Platycaria, with alnos. They host the more exclusive kind of DTs. So this is one of answers maybe. And the other one that these 3 morphotypes actually are the most common in each area. So the dicot tree is the most common. Lygodium is not a dicot, so it's not in this analysis. So dicot for the edge, Platycaria from the interior, and alnos from the bighorn. So what this is showing us that maybe because these plants are so available, they're so frequent, they're there. They're really abundant kind of resource for insects. So maybe this brings different kinds of insects eating or eating differently these plants that are so available. And plants that are less available maybe have more generalist kind of insects eating. This is probably what's happening here. Okay, so about the richness of damage. We want to see what area is more rich, more diverse in relation of damage. How many, how can I say that? You know, yeah, if you get this analysis, say like that, I have like 100 leaves. If I get 10, randomly 10 leaves, and I look for the damage, I will have a result. And then I resample more 10, and then I resample more 10, and I will compare all, the results all the time. I will see who is more rich, or what the combination, who gives me more DTs in different combinations. So we are fair, because as I showed you before, I don't have the same number of leaves in all these areas. So how I will compare like 29 damage in 900 leaves, it's more or less a third of 40 damage in 2,000 leaves. It's not a fair comparison. So we have to do this to have a more realistic kind of comparison. And this shows us that it's really, really also unusual that the most rich for the damage is the bighorn. And Bighorn Basin is the less diverse flora. So the less diverse flora has the most rich damage assemblage, let's say that. And usually it's more linear. As richer and diverse is your flora, more insects and more kind of damage you will find in this flora. So this was really, really unusual. So in blue, as I told you, is bighorn with more types of damage. followed by the interior of Pinheiro, and the last in relation to herbivory, with last types of herbivory, is the Pinheiro edge. So it's the opposite that we were expecting. When we look at just about specialized kind of damage, like mines and galls, we have the same pattern. So Bighorn has more types of mines and galls, followed by Pinheiro interior and Pinheiro edge. And as I told you before, we share in the three areas just two morphotypes, and we decide to check how is herbivory in these morphotypes. As I told you before, if the same morphotype in different areas behave differently, and how fits with the bigger view. So first with dicot tree, we can see the dicot tree does not fit the general pattern. So we have the dicot tree has more damage, more types of damage in the interior of Pinheiro, not in Bighorn. And popples as follow the pattern. So more type of damage in Bighorn, followed by the interior and then the edge. So this is showing us, mostly the first one, that yes, probably we have different faunas acting in these areas. So like the Pinheiro, we have like some group of arthropods acting in the plants. Pinheiro interior, we have other group, and Bighorn other group. So that endemism in plants will be reflected in the herbivores and the food webs. Okay. And here, this is how the sites are in relation to the functional feeding groups. So it's not about damage diversity, but as I told you about the groups. So who eats doing holes, who eats doing mines, who eats doing oviposition, who eats doing position of, not make sense, but who does oviposition. So here we see same kind of, that we saw for the flora. Oh, sorry. We have Pinheiro edge, separately from the Pinheiro interior, separately from the Bighorn, and Bighorn and interior has some overlapping in relation how they are eating by insects. So as I told you, this is the major conclusion here. These three distinct flora were eaten by three distinct kinds of insects, of community of insects. So we have different kinds of food webs happening here. So, Pinheiro less diverse by the contrary that we were expecting. Bighorn more diverse in relation of damage. Bighorn interior, again, more similar. They have been eaten more similar if we compare with the edge. And here we think that the specialized kind of damage has a huge impact in these results. When we put all this together, what we can see about Wyoming ecosystems during Eocene, and maybe what ideas this will give us about our future in the global warming. So Wyoming was much warmer and wet than it is today, for sure, as we saw in the beginning. This hot house that we have during Paleogene with the PTM and ECO, so we are expecting that this more homogeneous kind of temperature, it will give us a more homogeneous kind of ecosystems and floristic composition. This is not what we are finding. This is not what is happening here. So we could think we have examples of endemisms here, and we are looking for factors that are contributing for this. So probably this difference in flora are because of paleoelevation. We know the elevation from Pinheiro, the edge, but we are still looking for data from the other localities, but we really think that we have difference because the Pinheiro edge is closer to the volcano, for example, and the others are more in the floodplain, probably they are more low elevation. Soil depth for sure, Pinheiro has this more moist kind of soil by the presence of lygodium. And as I show you, we have 3 different floristic compositions. We have 3 different ecosystems acting there. And these plants are related with different herbivore, with different kind of consumers, as we could see here. So, in a global warming future, what we could predict or maybe or think about it, this will interfere in our food webs for sure. Yeah, the higher temperature will change the rates of herbivory. Usually when we have higher temperatures, we have more insects eating plants and eating in more ways. It's also related with pressure of CO2 and O2. So maybe we'll have higher rates of herbivory in our future. But the hothouse, the absence of ice in the poles, it means that we have a homogeneous kind of landscape? Not necessarily. Because as I can show you, paleoelevation, depth, other variables, even like sun light can be a major aspect in the floristic composition. Okay, and of course, as I told you in the beginning, the age. Here we have 2 million years old, 2 or 3 million years old from this guy, from these 2 guys, and maybe this is why they are more similar between each other. But the trend in this worm is the same, doesn't matter the age. Okay, so I think this is this is what I want to talk to you today. This is more or less the major results of my postdoc. And in the near future, we will include the data from Bighorn Basin and Hanna Basin. So we have a bigger picture about this endemism and about the distribution. Will be really interesting to see. And I have some fossils for you when you want to come and talk about it. And so thank you. I want to thank you first, Dr. Furano. Unfortunately, she could not be here with you guys, but I'm sure she really wants to come. She's super excited to come to Jackson and talk with you in the future. So this is people that helped me with the analysis and ideas and morphotyping at the museum that we're working. They're really grateful. I'm supposed to be working there today. Dr. Laura Vietti released me to be here. My funding, NSF, CNPq from Brazil, the University of Wyoming, and Dr. Curano actually started this project in Miami University. And of course, John and Mike that helped me coming here, all the emails, all the patients, and thank you. Okay, I don't know how it works.
Well, we have time for questions.
Questions?
Yeah.
Or, yeah, okay. Questions?
So time for both. So I mean, looking at the fossils
Yeah.
and questions. So let's do questions first.
Okay. Sure. Hi.
I'm trying to picture from your talk what things look like in the Eocene era.
Oh, I forgot to tell, it was a tropical forest.
Oh, it was?
Yeah, I'm so sorry. Yeah, I forgot to put the word.
With lots
Yeah, it was a tropical forest for sure. A different kind of tropical forest, but totally tropical. With not tropical Brazil, but tropical Florida. You know, with totally tropical.
With lots of insects or just some insects. If I were to go hiking in the Bighorns in the Eocene era and sat down on a rock, would I be inundated by insects? Or would I just see a few?
No, I think it's like to go, I don't know, to Texas during summer. It's it's a lot, yeah. We have some associations, some work showing the higher number of deities reflect the number of species. So that if you have like 39, 40 deities in comparison to 20, the 39 and 40 area, you have more insects for sure. Yeah, even if it's not directly, because one insect can bite in several different ways. So, did I answer? That was tropical, totally tropical. Yes,
Is there a fossil record of the insects?
Not here. Not here, unfortunately. Usually this is a problem, actually. Where you find leaves, it's really, really rare to find insects. The type of fossilization, the minerals, I think are different. Yeah, no, we don't have. At least not in Pinheiro, for sure. Bighorn maybe, but I don't think for these formations, no. Unfortunately, yeah. Who was with their hand? Yeah.
Would there have been seasonal changes, although it was tropical?
Yeah. With the technology, with the knowledge that we have today, by the plants, we cannot talk about seasonality, unfortunately. But we are aware that probably we have a more wet and more drying season. And when you talk, we put all in the mean annual temperature or the mean annual precipitation. We are putting a lot of error, a lot of problems there. Like, because you have a mean annual, a mean temperature for winter, a mean temperature for summer, that even for Wyoming, I was researcher to talk about, it changed a lot. So, yeah, some papers talk about the proportion of teeth and unteeth and lobe and unlobe to see seasonality, but they are not like a really good tool for now. Yeah. Hi.
So can you talk a little bit about, when I think of a forest with lots of insects,
Mhm.
the leaves fall down and then they get decomposed
Mhm.
and chewed up, but obviously you found all these fossils. Can you talk a little bit about how and why these were preserved?
Oh yes. Yeah, all these leaves are for sure in the floor or in the lake. These, more specifically from Pinheiro, they have volcanoclastic sediment. So probably the deposition was fast, and this is why they were not decomposed or eaten by or fungus and other things. One common question that we have, and how we know that it's an insect and it was not because it's dead and has a mark. And as I told you, because these reaction tissues, I can show you later, we just seeing live plants. If an insect eats a dead plant, it will not leave a mark. You get the hole, but you don't have that arrow around the row. So this is why how we know if the plant was alive or dead. But yeah, to have all these leaves, we have a more faster kind of was covered by the sediment.
Very
Yeah,
well.
yeah. Thank you very. Yeah. Hi.
As you were talking, I was trying to visualize if I were walking from the edge of the Wind River Basin into the interior
Yes.
and then walked up to the Bighorn Basin.
Yes.
What would really be different? You know, which is wetter, which is
Yes.
shadier, what-
So we think that let me put on that just to make some, no, okay, okay. Well, I think here, this is an old map, but I still use because I really like. So, here in the edge, we are close to the volcano. So the volcano more or less here. So you're in a high paleo altitude, and we have all this influence of the ashes. And here at the Eocene, we used to have like a river coming and crossing, and so the interior of this basin actually is the sediment from the floodplain of this river that are coming from up north. So usually, water comes from the high to the down. So we know that here is a little bit lower for sure. And these are different kind of rivers. We have more meander, yeah, we have like the sinuous kind of river coming here. So probably also was more low elevation. And when you have like sinuous kind of river, the physiognomic can change more faster, because sometimes you have an overbank or something and change the river path. And this is also have a fat burial and have a different kind of plants associated to this more unstable environment, because a meandering river, the vegetation around must be more adapted to change really fast. So yeah, this probably were more stable environment in comparison with these and these. So, but they are tropical forests. Hi.
Insofar as climate has a lot of variables, moisture, elevation,
Yeah.
temperature, oxygen,
Yeah.
how can you, the only thing you're really talking about here is temperature.
Yes, because to do isotopes, you need organic matter from your leaves. And we don't have enough to do isotope analysis. Will be great. This is one in Hanna Basin. One of our grad students actually is looking for this, to have the isotopes to put the information together. So we don't have like local. We use like a more general, like Zakos kind of curves for this age, for these areas, but we don't have like local data, because temperature we measure with the leaves. So we know that, of course, we know it's not perfect, but we have idea the temperature in that location, but because of the leaves, because of the morphology of the leaves. Because, and because we don't have this organic matter to do the isotopes, we don't have the data for that site. So we have to use more regional data.
So what would the other variables be in order to be predictive of our warming trend, let's say 200 years in the future? What other variables do you need to make a reasonable interpretation of a projection?
Other variables. Well, yeah, temperature and CO2 and oxygen, we already have really good kind of predictions, but not from plants. Really not from plants. And I think honestly, I think they're the most important variables, atmospheric variables, to make predictions in good and reliable models. But from the plants, I think you can do model about the type of ecosystem that you have. If you get drier and colder, what kind of more actual example, if Wyoming start to get warm again, probably the tropical or the more tropical forests from the south will start to come up, and maybe in 3, 4, or probably more age, we will have a more warm and wet again here in Wyoming with a total different type of vegetation. Yes.
Why would you say it would get wetter? Because we are basically a desert in so many ways now.
Yes.
I mean,
No,
but the other variable
if
is
we were.
moisture.
Yeah, actually, for Wyoming, I don't know the exactly predictions, but in a short way, it's getting wetter. It's getting more moist, like in precipitation. But when you look the vegetation, actually we have more like the desert kind of vegetation. So honestly, I think we are not there yet to do modeling. We need to study more, because usually when we talk about climate change, everybody expects that maybe the general view will be hot. Not necessarily. I think for sure some places will be more dry and more cold. But yeah, it's more complicated than we...
Because the temperature, you might be able to generalize about
Yeah.
temperature, but not about moisture. And that's a key thing, because if
Yeah.
you don't get that here,
Yeah, because the precipitation, you can predict precipitation, but you cannot predict how this water will stay in the system, for sure. Yeah, we can use some variables like plants with nitrogen association in their roots. This helps to keep moisturized, or fungal association, for example. But yes, we have a speaker just about global change, so probably maybe they have more, help you with this answer. Yeah. No?
So if there are no more questions,
Yeah.
we'll have some time to look at the absolutely stunning fossils.
Yeah, maybe you can remember,
And,
right?